So, dear learners, coming to part 2. After learning about the sources and action of amylases, we shall now consider the nature and physiological implications of amylase inhibitors. Alpha amylase inhibitors are substances that bind to the alpha amylases and making them inactive. Microorganisms, higher plants and animals, they produce a large number of different protein inhibitors of alpha amylases in order to regulate the activity of these enzymes. These inhibitors can be grouped into 6 classes as indicated by Richardson based on their tertiary structures. Class 1 is Naughton type alpha amylase inhibitor. This is the smallest proteinaceous alpha amylase inhibitor. The inhibitor from amaranth seed, which consists of only 32 amino acid residues with 3 disulfide bonds. The second is Kunitz like alpha amylase inhibitor. This contains 180 amino acid residues with 4 cysteines. They are present in cereals such as barley, wheat and rice. Third type is cereal type alpha amylase inhibitors. These contain around 120 to 160 amino acid residues and they have 5 disulfide bonds in the structure. They are present in wheat, barley, rye and ragi. Ragi is the Indian finger millet. These inhibitors are bifunctional. That means, they inhibit mammalian, insect and bacterial alpha amylases. Gamma thionine like inhibitors. These alpha amylase inhibitors contain only 47 amino acid residues. They are sulfur rich and form a part of gamma thionine like superfamily. They are present in sorghum, papaya, cowpea, wheat and barley. Next is thaumatin like inhibitors. Zeaxanthin and alpha amylase trypsin inhibitors are proteins isolated from maize and barley respectively. These proteins are homologous to the sweet protein thaumatin and the pathogenesis related proteins. Thaumatin like alpha amylase inhibitors contain 173 to 235 amino acid residues and they have around 5 to 6 disulfide bonds. Next is lectin like alpha amylase inhibitors. They contain 240 to 250 amino acid residues and they have 5 disulfide bonds. These inhibitors are present in leguminous seeds such as common bean, scarlet runner bean and cowpea. In animals, alpha amylase inhibitors reduce the glucose peaks that can occur after a meal, slowing the speed with which alpha amylase can convert starch to simple sugars until the body can deal with it. This is of particular importance in people with diabetes, where low insulin levels prevent extracellular glucose from being cleared quickly from the blood. Therefore, diabetics tend to have low alpha amylase levels in order to keep glucose levels under control, except after taking insulin, which causes a rise in the pancreatic alpha amylase. Plants also use an amylase inhibitor as a defense strategy. These inhibitors impede the digestive action of alpha amylases and proteinases in the insect gut, thereby acting as insect antifeedant. As a result, alpha amylase inhibitors have a potential in various fields including crop protection and in the treatment of diabetes. Now, let us see the health benefits of alpha amylase inhibitors. In order to understand the health benefits of alpha amylase inhibitors, we shall first study the role of alpha amylase in the disorders of carbohydrate uptake that may cause several health problems such as diabetes, obesity and oral diseases. Diabetes mellitus is a metabolic disorder resulting from deficiency in insulin secretion, insulin action or both. This promotes disturbance of carbohydrate, fat and protein metabolism. The therapeutic strategies for the treatment of type 2 diabetes include reduction of the demand for insulin, stimulation of the endogenous insulin secretion, enhancement of the action of insulin at the target tissue, and the inhibition of degradation of oligosaccharides and disaccharides. The drugs commonly used in clinic to handle or control the diabetes are insulin, sulfonylureas, biguanidine, glucosidase inhibitors, aldose reductase inhibitors, thiazolidine diones, carbamyl methyl benzoic acid, insulin like growth factors, etc. The effect of these drugs is aimed to lower the level of blood glucose. One therapeutic approach for treating type 2 diabetes mellitus is to decrease the postprandial glucose level. This could be done by retarding the absorption of glucose through inhibition of carbohydrate hydrolyzing enzymes, which are present in the small intestine brush border, and they are responsible for the breakdown of oligosaccharides to disaccharides and ultimately into monosaccharides suitable for absorption. Inhibitors of these enzymes like acarbose delay carbohydrate digestion and prolong overall carbohydrate digestion time, causing a reduction in the rate of glucose absorption and consequently blunting the postprandial plasma glucose rise. Dental caries and periodontal diseases are the most prevalent oral infectious diseases that cause significant impact on person's overall health. In etiology of dental caries, the most abundant enzyme in human saliva, alpha amylase, possesses three distinct biological functions in the oral cavity. First, its hydrolytic activity is responsible for initial breakdown of starch to oligosaccharides. Second, several lines of evidence indicate that salivary alpha amylase bound to tooth enamel or hydroxyapatite may play a role in dental plaque formation. Third, alpha amylase in solution binds with high affinity to viridans oral streptococci, and bacteria bound alpha amylase is capable of hydrolyzing starch to glucose, which can be used as a food source and then further metabolized to lactic acid. Localized acid production by bacteria can lead to dissolution of tooth enamel, a critical step in dental caries progression. Inhibition of alpha amylase enzyme that plays a role in digestion of starch and the glycogen is considered as a strategy for treatment of disorders in carbohydrate uptake such as diabetes, obesity as well as dental caries and periodontal diseases. Plants are the important source of chemical constituents with potential for inhibition of alpha amylase and can be used as therapeutic or functional food sources. The third situation is obesity. Overweight, obesity and diabetes are most common disorders associated with each other, and they are the major problem in the modern world. In most diets, carbohydrates are the greatest source of calories. Inhibition of carbohydrate digestion or absorption can decrease the calorie intake to promote weight loss and combat obesity. It is also a mechanism for reducing hyperglycemia in diabetic subjects. Before being absorbed by the body, carbohydrates must be broken down into monosaccharides, and alpha amylase catalyzes the hydrolysis of alpha 1,4 D-glycoside linkage of the starch and other glucose polymers. Inhibitor of this enzyme can be used in treatment of obesity and diabetes. A method of curing and preventing obesity comprises orally administering alpha amylase inhibitors, thereby inhibiting alpha amylase activity in saliva and pancreatic juice and reducing the digestion and absorption of starch, thereby reducing the calorie intake from the meals. Obesity can be cured or prevented effectively while taking usual diet without giving any physical or mental pain. So, students, in this part we have studied the nature of amylases, the physiological implications of amylase inhibitors and their prospective use in treating different conditions like diabetes, dental caries and obesity.